Hello, everyone, and welcome to today's session on high throughput omics data processing. Today, we are going to learn about transcriptomics data and what we did in the last class. So let's brush up with the basics. Let me start with a question. So, what is transcriptomics? Transcriptomics is the study of RNA sequences, copies of selected regions of genome which transcribed in order to make proteins or carry out other functions, right? That is that is transcriptomics. We are studying the RNA sequences that transcribe and make protein for different functions. Transcription is a dynamic process. We all know that, right? That transcription is a dynamic process because it keeps on happening all the time. So the transcriptome would be a snapshot of genes transcribed in a cell at a particular time, right? So what I'm trying to tell you here, for example, transcription, which is happening right now with me, with you. If someone comes and collects my sample right now and do the transcriptomics analysis, that would be for this time, that 8:33, 20th of January is the time where they took the sample. So that would describe my transcriptome at that given point of time, right? Or your transcriptome at that given point of time. So it's a snapshot. It does not tell everything about every state. It just tells about that particular state because it's a dynamic process. It's happening all the time. The sum total of RNA in a cell at a given point is the transcriptome. So what it suggests, what it says, the transcriptome is the RNA which is there in the cell at that given moment. In the last meeting, when we all met, we discussed the data generation techniques and how NGS data is processed. We discussed the data-driven hypothesis or discovery and how transcriptomics or RNA-seq data is generated, processed, and aligned. We talked about mapping reads, genes and isoforms, and different types of file formats. So today, I wanted to, recap. So let's again recap the different methods to measure mRNA. So there are various methods available to detect and quantify expression. So when I'm saying expression, which means that it's gene expression. So we are talking about RNA-seq, and we are also talking about expression of gene or a protein, and how to get those, how to use those methods to get the result. So in this figure, you can see reverse transcription PCR or RT-PCR amplifies DNA based on 2' sequences framing the section of interest. Since PCR amplifies DNA, not RNA, RNA is first converted back to DNA prior to amplification, right? So this is done via a process called reverse transcription. I think many of you have heard about it. And in nature, this process is carried out by retroviruses. Another method is northern blotting. So in northern blotting, an RNA sample is digested into fragments. Then they are separated by size. As you see in this gel image, that you see different sizes. So what is happening is that the RNA samples are digested into fragments, and then they are separated by size using gel electrophoresis. Then they are transferred to a membrane that can probe or test the presence of RNA or the RNA transcript. Another method, which was another very popular method, is microarrays. So, like northern blots, microarrays are hybridization based. But in contrast to northern blot, a microarray yields highly detailed information about individual genes that have been transcribed. So in this image, you can see different colors which represent a particular gene. Each color represents one gene and its expression. For example, if you see the green images, green dots, the more darker they are, the higher their expression. So, NGS technologies capture DNA sequences that we know and generates digital data using several steps. So, in this link shared with this image, you can read more about it and learn more about it. But to give you a brief understanding, first, what we did, first RNA was extracted from cells, and then it was converted into cDNA. And then it was sheared and placed in a flow cell. So inside a flow cell, cDNA fragments are amplified using bridge PCR amplification, and the flow cell is then inserted into the sequencer. So this is the typical process of data generation. Then the machine, the sequencer, uses image analysis to capture each letters in the flow cell fragment, in the flow cell fragments, by analyzing their visual patterns and converting them into sequence of letters. So from your samples, the biological sample is now converted into letters that you can read. So typically, NGS reads are between 30 and 300 base pairs long. And as you see, they consist of series of letters like TCGA. Then what the next step is to process or to start processing the data that you got from the machine. So to process next generation sequencing data, we must map those reads to a reference genome, right? To do so, we have to know where on the genome we have exons. So in the last class, Dr. Prabuddha told us a lot about the DNA and the regions that code for the DNA, the exons and the introns. Dr. Prabuddha also said that only 2% of the genome codes for proteins. So those are coding regions or exons. So as I was telling you, so we have to find those exons, right? Because we have a big genome. Just imagine that like only 2% of exons, and they are distributed all over the genome. And now what we have to do is to find those exons and map them to the reads that we have obtained from the sequencer. So a GTF file, here you can see a GTF file. A GTF file has that information about the genome. And the FASTQ files are the raw reads that we got from the sequencer, which has information about the short reads. So that the next step would be to align those and get the information, right? The genome reference gives us information on where the gene is located and where the exons are in that on that gene. So uh, then we also talked about a concept about the isoforms. So we'll talk in more detail by looking at this image. So as I was telling you that different isoforms can be created from a single gene. In this example, you see there is one gene. From this gene, through alternate splicing, which we discussed in the last class as well, and the codons and how they work. So I'm not going into much detail again here, but a single gene can produce different isoforms. So isoform 1 is a different protein, isoform is a 2 is a different protein, but it is transcribed from a single gene through alternate splicing. So to capture that process, to capture isoforms, we then use different reads which are mapped onto specific exons. Because they are mapped in different quantities, FASTQ files that gives different sequences for these reads. As you can see in this example, here on chromosome 15, you have the information that on chromosome 15, there's this exon, and this is the length from which it starts, and this is its end length. So it describes the particular location of that exon. Also, it also gives like gene and transcript ID, etc., etc. So this was the review that we did in the last class. Today, we are going to learn about NGS data analysis and different algorithms that are used to make this raw data into information that makes sense. So what we are trying to do here is that with the NGS data that we are generating, from the samples to the reads, we are trying to process that data. Why? Because while doing these experiments, they have their own limitations. Because in some of the cases, like in ribosomes, we have to put some adapters so that those sequences can be identified. And to remove those adapters, we have to use specialized algorithms that can do that. So we will be learning about those in this class, and also we will be learning about how to build those logical pipelines with which we can do the data analysis. So what we are trying to do here is we are trying to process the raw data and make sense out of it by generating a table of expression. So from raw data to a table of expression which has biological meaning, but a raw data with a lot of uh, artifacts, a lot of noise is not the data that you want to go with without processing. So pre-processing is very important, as well as mapping is again very important, and that too with the annotated genome. So we'll be talking about all those in this class. So RNA-seq analysis project. We are going to learn RNA-seq data analysis using examples, because I believe that with example, with the case study, with the biological question, we'll be able to relate the technology, and we will be able to understand its application as well. So we will try to do that in this session. So let's first try and understand the experiments that are required to do a RNA-seq analysis project. The outcome, or the understanding that we will be getting out of it, is that how we can think about a RNA-seq analysis project. What are the basics? So the first thing is defining how many groups are there in a given experiment. So what are groups? Why we are talking about groups here? We are talking about groups here because we are doing a comparative analysis. When you are aligning your your transcriptome, for example, with a reference genome, you're comparing, right? So what we are trying to do here is to do a lot of data comparison from different samples. So defining groups typically starts by looking at the conditions. Sometimes, for example, it could be treated or versus controls. For example, someone has been treated with a drug versus someone who has not been treated with a drug. So this is like two different groups, right? So as a result of the condition or groups, we can identify different patterns. So that is another important objective. Sometimes we don't know about the conditions or the groups. In those cases, we can try to identify those groups by analyzing the data. So in this program, we will be going through all those steps. So the second is determining if an assembled genome is available and annotated. As I was telling you before, the genome is very important because when we have raw reads in FASTQ files, we need to align them to to a reference genome before we can study the genes or expressed isoform expression. Why? Because the genome or the proteome that you have sequenced, you don't know anything about it. And to do a comparative analysis, it would be great to do it with a known genome where you know the positions of the exons, where you know the positions of the genes. That helps us to reduce the error. And lastly, what is the objective of the analysis? So this is very important because the objective of the analysis could also be your biological question, right? So how to understand or I make a good objective of the analysis? So objective of the analysis can be determined by having clear comparison that has to be made. As I said, that here we are typically doing some sort of comparison. So we should have a clear idea about what sort of comparison we would want to do. Usually, I mean, typically, these sort of comparisons are, to find a gene, is to find a gene. Maybe we are looking at number of genes that we want to find, or a feature. For example, that we want to find a feature that helps us in, classifying our samples so that we can do the comparison. So these sort of things are the objectives of us would be study. After defining the objectives, we would go into the practical example. The data set. The data set is very critical, and I think it's an important slide here for everyone to be a little focused. The data set we will be using is educational data set based on public domain data from a publication by an author called Bradford et al. 2016. So we will be sharing this publication with you in the next email. So this study or the data which is used in the study is also used in one of our projects, which is called patient-derived xenografts, transcriptomic biomarkers for breast cancer. So we will also be sharing you the link of that project. In this example, we will be using methods for RNA-seq analysis. So in this publication, what had happened is that researchers from the study investigated samples from 3 different types of breast cancers. So they did it because they want to better understand and characterize tumors. So that was the purpose. They took they were investigating these samples that has like different types of breast cancers. So there are different types of breast cancers which falls into different categories. So I will be telling you more about that. But to understand this, what they were trying to do is that they wanted to characterize these tumors and as well as their microenvironment. So this model was helpful because here we are dealing with 2 distinct tissues coming from 2 different organisms. So as I was telling you that I would explain more about these types of tumors. So three different types of breast cancer tumors that were implanted in the mice here, as you can see in the mice, are ER positive, which is estrogen receptor positive, HER2 positive, which is human epidermal growth factor positive, and triple negative. So triple negative means negative expression for ER, HER2, and progesterone receptors. So these are biomarkers which tells you about whether the presence of a particular tumor or not. So if someone is triple negative, which that means that that person does not have it. Okay. So from the full data set, we will only be taking two subtypes of breast cancer in this example, which is ER positive and true negative. In the full project, which I discussed before, also I'll show you again. There are 7 samples and 7 ER samples and 13 TN samples. So in each type of breast cancer, they have different characteristics, which we will be studying through gene expression. So that is the objective. For the full data set, we will take only 2 data sets, as I said, which in these 2 data sets, 7 samples of ER positive and 13 samples of TN negative. Okay. Okay, now let's define some of the more objectives in more detail. So talking about the samples, we talked about the samples, and we also want to talk about the reference genome, because that is also important. The project samples were taken from humanized mice, which is, I think it is well described in that project. So you can go and check it out. And the implanted tumor from human patients. So from the human patients, the tumors were implanted into the mice. So as a result, we have two different assemblies, one from the human, one from the mice. Two different assemblies and annotated genomes to map the RNA reads. So we have to use a genome assembly which has both both human genes and mice genes as well. So this will be our data set. In this project, we will be looking into the expressed genes and isoforms, because those mRNA will become proteins. This means we will not be also looking at non-coding RNAs and isoforms, because because by not taking them, this simplifies our analysis and allows us to focus on higher level of variation. So another objective of this doing this is to find genes associated with functional processes, typically through breast cancer subtypes. So the breast cancer subtypes that we described. So we are trying to find genes associated with them. So let's see how we can practically see the various aspects of RNA-seq pipeline using the data set. And the project, as I was telling you, that the project is here and the publication is here, but we'll be sending you through email as well. This is the project which describes this analysis and the publication link. So I think let's take a break. And if you guys have any questions so far from the slides that I've shown and the things that we discussed in the previous class and that we reviewed in this class, so please type in, and you can also ask me. Okay, as we don't have any questions, so let's move on because we will be doing a lot of things today. So there's one chat. Okay. Yeah, meanwhile, I can actually do that. I can post the links in the chat. That's a good idea, Bibsha. Okay. So it's not happening. So I'll share the links anyways. We'll be emailing you those. So it's the project in the Edu platform, and also the link is there in Transcriptomics 1. So anyone who goes through Transcriptomics 1 will be again going through the concepts that we are going through today. So in case, I mean, you don't understand anything, you can totally go back to Transcriptomics 1 and learn all of the things that I've told you today, so that there is a book so which you can refer to, which is much more detailed. So moving on, we were discussing the pipeline, which we are going to do next. Before we do that, let's just recap some of the things that we did last time again. So an RNA-seq pipeline typically includes three main steps. One is pre-processing, another is mapping, and another one is quantification of expression levels. So pre-processing is needed to clean up our data. I told you we discussed a lot of times that pre-processing helps in removing the adapters, trimming some of the reads, and removing the PCR duplicates. And then we take the cleaned up read sequences to map them on a reference genome using the FASTA files for sequences and GTF FASTA or FASTQ files and GTF file for annotation. So based on the quality of the annotation, we can use various strategies for mapping. So quality is one of the important aspect, and the quality is also determined by the level of annotation and all those things that we said, and the sequencing depth. There are multiple things which you can totally read in Transcriptomics 1 and ask us as well. So once this step is complete, we have to quantify expression levels and give each of the expressed element a number. So that is very important. So this whole process, I mean, many people were asking about what is a pipeline. I think it's something that we should say that what is a pipeline. So this process is called a pipeline. A pipeline is a workflow that has interconnecting steps where each next steps takes the output of the previous step. So this step will take the input from this. So from here to here, from here to here. So each next step takes the output of the previous step. The workflows utilize certain file formats that are structured data in a stepwise manner. Like the file formats are taken care of in a pipeline, where you have to take care of those in a pipeline where you are fixing errors in the read, cleaning up unwanted technical variations in the pre-processing stage, and then mapping onto the genome on transcriptome, and after that structuring the raw reads into gene and isoform. So during all these processes, you have to take care of the file format as well. So these all are done in a pipeline. So that is a pipeline. So we also discussed in the last class about these algorithms, PCR-clean and Trimmomatic, and also we discussed about the mapping on a reference genome, which is in which we use, I think, Bowtie 2, which is a fast alignment algorithm. And the logic behind it is that it's based on seed approach or a KMR approach. I think you can read more about it in the reference publication, which we will be also showing in the pipeline. And another is TopHat. So TopHat is a fast splice junction mapper for short reads. So TopHat aligns RNA sequences reads to a reference genome using Bowtie. So Bowtie is a short read aligner. Bowtie is a very popular algorithm, and it is based on Burroughs Wheeler index, and I think you can read more about it in the publication. But I mean, after the reads are aligned by Bowtie, TopHat analyzes the mapping results to identify those splice junctions between exons that we showed you previously as well. So another is HiSat 2. So HiSat 2 is an alignment algorithm alternate to TopHat 2. So similar to TopHat and TopHat 2, HiSat 2 aligns read to the reference genome and builds a map of junctions reads for assembling transcripts. So these all things we covered in the last meeting, and also we did a demo pipeline. So mapping methods, as you see, these are the popular mapping methods, and they're color coded in red. So now I think for doing the next step, which is which is very important, and which is to after pre-processing and the mapping step, we wanted to do the expression values, look into the expression values for genes and isoforms, which are generated through an expression table. So we'll be looking at the expression table. So this is usually done by an algorithm called RSEM. So RNA sequence by expectation maximization is the name of this algorithm. So RSEM, which gives us result in RPKM and FPKM and TPM. So let's go to the server and let's let's run this and check out the results. So I think everyone here has the login credentials now, and if we can all log in and start doing the analysis together. How about that? So here you can see I have already logged in, so I'll log out, and if you open this URL, okay, there's a question, or a good question, I think. So this is the link. Please go to this link and let's all, all of you, let's go to this page and open this page, please. And then if you are there in this page, please write yes in the chat box. So meanwhile, let me answer the question asked by Srijini. What is the difference between mapping and annotation? It's a very good question, Srijini. Thanks for asking. So in mapping, what is happening is that the short reads that were, that came through the sequencer are aligned to the exons. In our case, we are looking at the alignment where the all those exons and introns where these map reads are matched, they are being aligned there. So they have been aligned at those specific positions where they have been matched. So that's the mapping. After mapping is done in the entire genome or the transcriptome, where all the sequences have been placed, next step is to find out what is this. So to find out what is this is the step of annotation, where you find out about that particular gene. Okay, this part is the exon part, this part is the gene part, because you could map it with a reference genome. Does that answer your question, Srijani? Okay, great. So only two people have said yes here. How about the rest? I mean, can you guys not been able to come to this page? Server.t-bio.info. Please try to log in. If you're using your mobile, I think then it will be problematic. If you're using your laptop, then I think you can easily access this page. And it would be important because we will be running the analysis, doing the pipelines together. Okay, so I think I will just start with. So yeah, we have now few more. So 5 people have been able to do it. So this is great. Thank you guys. So it's pretty simple. You will understand once you start accessing it. And today we are, as we discussed the um, project and the data in that project, we will be doing that. So here in this section, NGS data section, so we have several sections as we described you before as well, that we have 4 sections here, and we are also coming up with newer sections. So we have NGS data, mass spec data, structural biology data, data integration and modeling. But today we'll be focusing on the NGS data part. So let's not go there elsewhere and get lost. Today we will be focusing on one pipeline, and let's just start slowly with one demo pipeline and try to understand how this demo pipeline are designed and why they are so helpful. Okay, I forgot to log in. So here, yes, so this is open. Let me go back again, just in case. I mean, here in this, if you click on this arrow, down arrow, you'll find 3 demo pipelines. And in the, we are going to the third one where we discuss the PDX modeling. Okay. Now we see the pre-processing section. So in our presentation, we saw this section, right? This is the section. We use PCR and Trimmomatic. So let's do that on the server. Here, we don't have to do anything, just click start. And in start, you can see a wonderful image which shows you different, algorithms and their how they work. So it's very easy to read this one, and it's also very graphical. So please do that once you practice it on your own. So here you have several links. So let's download this. There's just 2 buttons here. You can click on it. So it will download as links.svl. So let's try open it. So it will not open like this. So let's see where it has been downloaded. Here it has been downloaded. So I'll open it with any text editor, which I have to find now. Okay. So in Windows, you can open it with several text editors. So it has a link. This SVL file has a link which is linked to another file which has the links to our FASTQ files. So we also have references. So let's start. As we saw a couple of things like Trimmomatic and PCR-clean, and we also understood why they are used. So we'll click on PCR Trimmomatic first and PCR-clean. And I'm hoping that everyone here uh, are also being able to do it with me. So I'll slow down and I'll wait for you guys to, click on those buttons. So we are trying to construct our pipeline here, and we do it by clicking on the start button on the demo pipeline. Okay, so here, I mean, in transcriptomics analysis, there could be two strategies: mapping onto the genome and mapping onto the transcriptome. We can do both. And we also learned about the mapping algorithms here. Here, we discussed these. I hope you remember. So mapping on the set of known transcripts, mapping on genome, so and potential identification of noble transcripts. That's why we map on genome. This could be a combined strategy also where you do the both. So let's go back to the server. We will use, let's use HiSat 2. Let's use Bowtie, and then we can have the R expression table because we already discussed that. So when you right click here, it will tell you whether you want to go to the previous step. So this is very important to know. I mean, if you have clicked somewhere else and you want to do the different, you want to choose a different analysis, then you have to right click on the button and it will ask you, are you sure you want to jump to the previous step? So it's very simple. You are going back now. So we went back to the start button. Now we can start again. Trimmomatic. Let's refresh it. So from after doing pre-processing, we are aligning it to the transcripts, and then we are generating a table of expressions. Differential expression is something that we will be learning in the next class, which is another important aspect and very uh, it's been used widely in research. So we'll be covering those and some of the statistical techniques, which is very important to further analysis, to the further analysis. So let's end this. Okay. So we used a strategy like this. So you can see what you did in the output. And then if you see here, you have a list of files that been created. So FPKM and all these expression, quantification techniques. Let's go back and learn about those in a little bit more detail here. Sorry. Yeah. So if you see, like, this is an example of TPM here, but we have different methods. So we will describe them one by one. So the pipeline results are, very clearly organized. So here, I mean, this is the screenshot, but this is the actual pipeline where you can see. So they are organized by exons, isoforms, and genes. So exons here, isoforms here, and genes. So you have different table for them, and we'll be looking into those tables as well. We also see normalized, and filtered table of genes and isoforms. So this normalized, how they have been normalized, and also we see mapping statistics file here. So these are the different files that are there for you to explore. So let's talk about the techniques. So expression levels can be quantified using algorithms that align reads. The whole thing that we saw, that the alignment to a transcript sequences. So HT-seq is another algorithm, and also RSEM, which we discussed. They are used to give this result for an expression table, and it is color coded in red. So about these normalization techniques. So RNA-seq data, I mean, produces abundance of transcripts in the form of read counts. Okay, so from the raw data, I mean, there's a lot of data that comes out. So for one individual, I mean, one position, there could be multiple reads. Okay, so but these read counts, they needed to be normalized to remove the effect of gene length and sequencing depth. So the gene length and the sequencing depth are the factors that changes the biological meaning. So what we are trying to do here, we are trying to do basic mathematical normalization, which could help us in making the data more sense in a biological aspect, or what is actually happening in that state where we took the transcriptome. So to do that, we do different types of normalization, mathematical normalization. This is the formula, and it is pretty straightforward. As you can see, FPKM, which stands for fragment per kilobase per million mapped reads, and it's divided by total number of mapped reads in millions into gene length in kilobase. Because as I said, gene length has an effect, and also the sequencing depth. You can read more about these normalization techniques in the link. Another aspect after doing the normalization. So let's look at let's just look at one of these files. So this is gene in fpkm.txt. So I just downloaded this file, and let me open this in Excel. Okay. See here, you see, this is the ID. So gene ID, which we discussed about the ensemble gene ID, and this is the sample, and this is the expression. That is what we are finding for so long. We were coming from doing all those steps to reach here, where we can see that at a given sample, what is the expression of a given gene. So in this table, you may notice that there are many zero values, and these zero values are of no good for us. For example, if an expression of a gene in every condition is zero, so that information, because there is no comparison, because everywhere it's the same, so we might not require that gene for our analysis. So we have to remove the zero values. So inside each cells, you will find quantified expression levels. So as here you will see that some of these numbers are very big and some of these numbers are very small. So to counter this, we do something called log transformation. And all those transformation techniques and the statistical techniques will be covered in the next class in our differential gene expression class, which is a part of transcriptomics too. So I think you can do this at your own laptop, or at your own convenient time also, this analysis, and you can check some of these results and try to understand that how this data correlates with the analysis which we did today. And also, I mean, if you see the transcriptomics one course, I mean, I think it would have been better if I had opened with something else. But yeah, maybe a Word file would have been past this better. So yeah, you can open these files into text editors and different editors and check the input outputs, which are also described in the course. But if you have any questions while doing this on your own, if you are not being able to do it, if you have any questions in any of the steps, if please reach out to us. We are all the time available, and during the sessions also you can ask those questions, but you have to go through some of the content. Another thing that I wanted to talk about is, let's go back to the presentation now. So this is the link, I mean, where you will be able to learn more about it. And as I was telling you about the linear scale versus the log scale, because we saw the linear scale, the numbers are, the relative differences are biologically more meaningful than absolute. So the comparisons, if you do, log scaling, does make more sense. So this is the expression table which we saw. This is another example. This is the sample name as you saw in the Excel sheet. And this is the gene ID. So we have, in that sample, if I go back again, sorry about that. So we have concatenated, okay. I think when we build our own pipeline, I'll show you the genome, reference genome. So just to give you an overview of that, I mean, once you understand these, you will be able to, create your own pipeline and run these analysis on our server. But to do that, I mean, first you have to understand the demo pipelines. So here, I mean, in this full version here, you will be able to, see the type of reference genome. So we have different type of assemblies, and also something that you can upload in a research license based query. Also the different type of file formats, and also the single end and pair end, and the organisms. So here you can see the last one, human mouse concatenated. So this was the assembly that was used in this pipeline. But I mean, because it's a demo pipeline and those parameters are already been set, so you don't have to do it. But once you understand those, we will be coming here and building our new pipeline, which will be running live on the server, where you can change some parameters and play with it and understand the output of the program. So that would be really interesting. And I think this part is, which we did today, is equally exciting, because we are from sequences, raw reads, raw data, we are coming to a table of expression where we can see that maybe here, if you start plotting this, we will understand the differences in gene expression of some of the genes which are there in some samples, and which some of the genes which are there in the other samples. So we'll be doing those things in the next class, and learning more from this data. But I think it would be great if you all can cover this much, and then start those analysis as well with me. But I mean, today I think to recap, we clicked here and then we went to the PDX project, and then we did the start, and then we did PCR-clean, Trimmomatic, and then we did Bowtie 2T, and then we did RSEM expression table, and then end. So that's how we created that pipeline, and also the outputs from those pipelines can be seen and downloaded, which we downloaded and saw. So next step is biological interpretation. So once you have the number of genes, which we will be discussing in the next classes about how we can, biologically interpret from those, sequences from those genes, how we can say which all are biomarkers, what the study that those researchers were doing. So we'll be repeating those things, and it will be more, biologically relevant once we go in more depth to single genes or number of genes. So if you have any questions, please write down in the chat box, or you can ask me as well. So we are coming to an end, and I would request, if you have your questions, please keep them coming in the box, chat box, or also you can unmute yourself and ask me. For this class, I think it would be great if you can start doing the transcriptomics one. So I heard that many of you had some issues and difficulties. Sorry, issues and difficulties finishing introduction to bioinformatics. But it's okay if you have not finished introduction to bioinformatics, you can start transcriptomics 1. So transcriptomics 1 is an independent course which will not require you to finish the introduction to bioinformatics course. But I think when you have leisure time, please finish introduction to bioinformatics as well, because it will yield you a very good certificate that you have gone through the introduction to bioinformatics, first of all. And second, that you will also understand what different application of bioinformatics, big data bioinformatics are there in the current scenario, which will help you decide your future better. So in this second assignment, we have another class on Thursday. So try to, it would be great if you can finish transcriptomics, but it is very elaborative and a great course. So I think it would be fine if you can just start with it and, go through the things that we discussed today, practice the demo pipeline, and then in the next class, we will teach you how you can build your own pipeline. And that would be really interesting. And along with the things that we did today, I think were really interesting. So please utilize the online resources, the edu platform that Biksha shared earlier, and also the on our WhatsApp group. I keep on sharing those links and those important publications. So whenever you have time to scroll through, please look into those messages and find the links which are important for you to go through. So you saw this image, which tells all about the different steps. So this is kind of a conclusion or the overview of the entire things that we did. And there is a link here. So if you can go to this link, it will give you the demo pipeline steps where all these algorithms have been used. So we'll be sharing this link as well. There's not much to talk about the user activity and progress lately, but that is all right. I guess all of you have been busy in some other work and trying to figure out how this all work. So please take your time, and we are here to help you. And as we promised earlier, that this would be a you know doing at your own ease kind of a thing. But it would be very good if you can do this along with me, along with our team, along with other people, so that we will learn more. But I mean, the good thing is that some of you are started doing it, and some of you have found it interesting. And I think in time, if you invest some more time, many of you will find this really interesting. So there are many courses. Some of the profile that stand out to me is this one. So if we can have like an image to the profile, it would be great, and so that we can see you, congratulate you. And also the names, if they are correct, please put them like not the username but your name that you use. so that the certificates have those names. And there are several courses that you can register into, and use these courses to enrich your knowledge using this program, because these courses are paid courses otherwise, but it would be made available for all of you. So please go through them in this duration of this course. So we have some upcoming workshops, which we always talk about the next programs. So in the next class, we will be discussing about the processing of NGS data and differential gene expression. As we said, that we will be talking a lot about statistical methods and how they are helping us, to make this data more structured and more towards making more sense. So we will also be talking about some of the machine learning method, like factor regression analysis, in the next class. We have upcoming class for differential gene expression tomorrow. So this will be another class on RNA-seq data and its analysis. So from this, I would like to conclude, and thank you everyone for joining today's meeting. I hope this was useful. And if you have any questions, please write to us in any form, through WhatsApp, through emails, through our LinkedIn page, through our Facebook page. So there are several mediums that you can contact us. Once you start utilizing it, I think there will be several questions. And also, please go to your university organization page, our forum, where we keep on, posting links, and you can see your own progress. So keep checking your profile. Thank you, everyone, and have a nice week ahead. We'll see you on Thursday.